Okay, now let us look into the applications of biotechnology in life. The first one that you need to know is gene therapy. Okay, gene therapy is used to treat or prevent genetic diseases. Okay, it is the treatment of genetic diseases by changing the genes in a person's cell. So the concept is that a normal gene is inserted into the patient to replace the abnormal gene. So examples of diseases that can be cured by gene therapy are cystic fibrosis, muscular dystrophy, and a few types of cancer. So this figure shows the basic steps in gene therapy. Okay, so first you are to insert a normal allele into a particular gene in the cells that contain non-functioning alleles of that gene. So if the inserted normal allele is expressed, then we say that the individual will produce a functioning protein, and therefore we say the individual no longer have the symptoms associated with genetic disease. Okay, so first, patients with genetic disease, their stem cells are isolated from the patient's bone marrow and it is grown in vitro. Okay, then the engineered viruses carry the normal allele into the host cells, and the cells that express normal alleles are then isolated and implanted back into the patient. Okay, so the second, so this is cystic fibrosis. Okay, the examples just now. Okay, now let's look at the second application of biotechnology in life, and that is DNA profiling. Okay, so these are the functions or the applications of DNA profiling, and that is to identify criminal, to settle paternity and maternity dispute, identify genetic diseases, and to determine suitability or the compatibility of organ donor and recipient. Okay, so this is a technique, DNA profiling is a technique used in forensic to identify individuals based on their DNA. And this is due to the fact that individuals' DNA is unique except between identical twins. And the human DNA set is different from animals. An individual can be identified based on DNA obtained from his or her blood, semen for male, or skin. So this is a figure that shows a few applications of DNA profiling, while this figure 13.6 shows the steps used in DNA profiling technique. Okay, so first, blood, semen, or skin sample is obtained from an investigation scene, for example, a crime scene. So next, DNA is extracted from the sample. Okay, the third step is that we are to use restriction enzymes to cut the DNA into different sizes of DNA fragments. So the DNA fragments of different sizes are then separated by gel electrophoresis. Gel electrophoresis separates DNA fragments according to size, where the large fragments will move slowly as compared to small fragments, and negative DNA fragments move to positive end. Okay, so the DNA fragments of different sizes, they are transferred from the gel to a nylon membrane, and the radioactive probes are added to the nylon membrane, and therefore X-ray film is then placed on top of the nylon membrane. Okay, next is that the X-ray film is processed to show the position of DNA bands that form the DNA profile. Okay, now this section over here. This shows the DNA profile, and from here, can you determine who is the biological father of the child? Okay, is it father 1 or father 2? Now let's compare these DNA fragments. Okay, so from here we can see 1, 2, 3, 4 of these DNA fragments of this child matched with father number 1. These 4 fragments, 1, 2, 3, and 4. Okay, as compared to father 2, only 1 and 2 fragments matched with the child, and therefore we can conclude that father 1 is the biological father of the child. Okay, so bio with laws. So DNA profiling can be used as a strong evidence in a criminal case if biological sample is obtained and processed correctly and consensusly. Okay, and if the forensic scientist tests the sample and analyzes it meticulously in a transparent manner. Okay, also the test results are interpreted precisely, and therefore the jury and the judge can obtain an accurate report. Okay, so this is a video on DNA profiling, which you can watch later by clicking into the link attached.